Okay guys, it's time to start on chapter 8, microbial metabolism. Hopefully for most of you guys, I know you guys have all done AMP1 or else you wouldn't be in this class. This will be a review, so it won't be totally strange to you like it was maybe the first time you heard it. So let's jump on in. Metabolism is all of the biochemistry that happens inside a living thing. Control biochemical reactions, ultimate function, reproduce. Well, actually, the ultimate function is to keep it alive. Reproduction is one of the things that it does. But all those chemical reactions are just the things that have to happen in order for life to exist, whether it's in a bacteria or a human being. Metabolic processes, 8 statements. Well, okay. Every cell requires nutrients. Everything needs food, guys. Metabolism requires energy from light or from catabolism of nutrients. So in order to do our biochemistry, we're going to need to have some energy. We can either get that energy from sunlight, like the plants and the algae do, or we get it by breaking down other molecules like glucose, for example. The energy that is released from these or manufactured is stored as ATP, adenosine triphosphate. ADP has that third phosphate group bonded on against all the repulsion of the like charges, and that requires energy, and that's exactly why we need energy to make that ATP. The cells are going to catabolize. Catabolism, I should have mentioned over there, that's the type of biochemical reactions in which you break something down from a big structure to its smaller components. Think if your children had a Lego play set and they had all these things put together, and you tell them, okay, it is time to go to bed or whatever. Take these apart, and as you take those Lego units off, that's catabolism. We do the same thing with all the big molecules that we come across, and so do the bacteria. Precursor metabolites, energy from ATP, and enzymes are used in anabolic. Anabolic is building up. That's taking those Legos and putting them back together and making something out of them. So we need the building blocks, whatever those building blocks are for proteins, for example, it's amino acids. We need energy because if you're going to put something together, you're going to spend energy. And most of the time, we need an enzyme that helps us to speed up the reaction significantly in order to put stuff together. And we got to put stuff together. Metabolism equals anabolism plus catabolism. That's what it is. Metabolism is all the stuff I break down and all the stuff I put back together. Those enzymes plus the ATP help us to form those macromolecules, and that's stuff like glycogen and again proteins. Cells grow by assembling macromolecules. We need proteins, we need all of these precursors, we need nucleic acids from nucleotides. It just makes sense, guys. Cells reproduce once they have doubled in size. So for the bacteria, once they have doubled in size, and of course they'll have to copy their DNA as well, then they're ready to reproduce by splitting in half or splitting into 2 separate organisms. Okay, we kind of already touched on this, but we'll go over it again. Catabolism and anabolism, those are the 2 types of metabolic reactions. Catabolic, break down, and these guys are exergonic. Ooh, that's a term. Terms always have a way of showing up on tests, don't they? Exergonic means we release energy. When we break something down, for example, that 4 molecules there, when they broke those bonds, it releases energy. anabolic pathways where we make large things from smaller things require energy. If I were going to put these guys back together again in an anabolic reaction, well, they've got it right there, then I'm going to have to add energy in. Endergonic is the term for adding energy in order to make a reaction happen. Okay, this is really good. Shows catabolism. We break apart whatever molecules that we use, usually carbohydrates or something like that. We break those apart. Part of it is just lost as heat. Nothing is 100% effective. Some of the energy is going to be stored in the ATP. We're going to take ADP right here, and using the energy released and another phosphate group, we're going to put it together. That is going to be used to put stuff together. Some of its energy is going to be lost in heat. And when we're finished, we've got these macromolecules, these huge molecules, and this is going to drive all the living processes of the cell, growth and division, etc. It's also going to be the way that we make the structures, membranes, ribosomes, bacterial cell wall components, phospholipids for the membrane. All of that stuff is going to be an anabolic process. This is how we classify organisms by where they get their energy and where they get their carbon. So let's make ourselves a table. Actually, didn't need that. Okay, so chemotrophs, these are guys that get their energy from chemicals like us. We get our energy from blood sugar or from lipids or from whatever organic molecules. We break them down. That's how we make our ATP. We are chemotrophs. Now, there's the chemoautotrophs and the chemoheterotrophs. Okay, these chemoautotrophs, they're all going to use chemicals. Sorry guys, lost my pen there. They're all going to use chemicals, the chemoheterotrophs and the chemoautotrophs. The difference is these guys have got to start with organic compounds. They can't make organic compounds from inorganic. These guys can get their carbon from an inorganic source and change it into an organic compound. Here you go. Let's keep these lines kind of straight. I know I'm not doing a real good job on that. Hydrogen, sulfur, iron, nitrogen, and carbon monoxide oxidizing bacteria. So they can use all of these different compounds, and they're able to convert these into organic molecules in order to use for their energy production. Chemoheterotrophs, chemical, but they have to have organic. So we've got two classes of these guys. Phototrophs. Photoautotrophs are going to use light to make energy molecules. A lot of them, like plants, glucose. The reason they're an autotroph is, again, they're going to use inorganic carbon, same guys, and they will do that. The photoheterotrophs, light, but they use organic compounds. That's not really too awfully hard, guys. So I would definitely know that for the next test. Redox is the way we call oxidation reduction reactions. A redox is where one atom or molecule gives up an electron to another. So the one that actually donates the electron is oxidized. The one who accepts the electron is reduced. And a lot of times in these guys, you'll see that these guys actually pass these off in a series. But for just understanding the redox reactions, there it is. From electron donor to an acceptor. And obviously these guys have to occur at the same time. You can't have an electron just floating around by itself. Every time that I have an oxidation reaction, I got to have a reduction reaction at the same time. Okay? Simultaneously. These guys are used to carry the electrons, and these are, if you guys recall, this is how we actually make the ATP. This is, in humans, NAD, nicotinamide adenine dinucleotide, is one of these electron acceptors. Remember, they pick up hydrogens or electrons or even hydrogen atoms from the glucose molecule and carry them over to be processed. We also talked about flavin adenine dinucleotide, FAD. Here's one we didn't talk about in human terms. It's going to be nicotinamide adenine dinucleotide phosphate, NADP. Bacteria are going to use these guys in the same way. They're going to use them to take those electrons from a energy source molecule such as glucose or fructose, lactose, and they're going to take them and instead of having a mitochondria, they're going to use the actual plasma membrane or cell membrane of their own cell in order to pump out hydrogen ion. Well, you remember. We don't go into all that detail. Release the energy stored in the bonds of certain molecules, and then we store these guys as ATP, or they do. Phosphorylation is when we take a phosphate group, which is a PO4, and usually we have 3 of these on ATP. Only the last one is the one that we use. And we add this to something, and by adding it, it actually releases energy when we break the bonds with the ATP molecule, and it's transferred the energy in order to power something. And you guys remember all this from A&P, so I don't want to have to try to repeat A&P. If you don't remember this stuff, then check your notes from A&P, or you can just shoot me questions and I'll be glad to answer them. But cells phosphorylate ADP to ATP in 3 ways. So this is how do we make it. There's one that's called substrate level phosphorylation. If you remember, that's what we do in glycolysis in human cells. We break down glucose and we're able to make some ATP directly. Not much, but some. Oxidative phosphorylation, that is what we do in the electron transport chain. That's where we use oxygen and we step down the energy levels of the electron, using that to pump out hydrogens, ions, and then allowing the hydrogen to come back in down their concentration gradient, and in the process they spin the ATP synthase. And then photophosphorylation, it's going to be more of a plant thing. And we use that energy by breaking that 3rd bond. We've already talked about that. Enzymes, again, this is rerun from A&P, guys. These guys are organic molecules, and these are catalysts. These are things that speed up reactions. How do they do it? By reducing the amount of energy needed to make it go forward. The reason I am not touring Europe on vacation is because it costs too much money. If they reduce the cost down to $100, I might be able to go, right? So it's the same thing. the cost of energy is too high to make these guys go fast. But when you lower the cost, the energy cost, then you speed up the process and you get more of the reactants coming together and making form of the products you're after. So the activation energy is how much it costs. There's the activation energy, and here it is lowered with a catalyst, and it just makes things go faster. We'll see another thing coming up too right now. Substrate, that's the thing that we work on. Now this could be a single substrate that we want to break into. This could be two different pieces that we want to bind together. The important thing is this guy is like a lock and a key. This is the key and this is the lock. The enzyme, the catalyst, has a shape that fits these guys. And by bringing them into close proximity, well, I'll just come over here like this, I can either break them apart if that's the purpose of the enzyme, or I can unite them if that's the purpose of the enzyme. Active site, if you look, these guys are kind of rounded and these guys are square. What happens is when these guys start to bind, it actually changes the conformation of this, and it actually changes to be a tighter fit. That's the active site modification. Enzymes and metabolisms. Okay, so Apoenzyme and Holoenzyme. Again, these are Anatomy 1 terms, but just in case you can't recall back that far. An apoenzyme is a protein structure that is going to be an enzyme, but it lacks something. It lacks another molecule that needs to bind in order for it to work. And this is a good control mechanism. You don't have this guy turned on all the time. You only turn him on when you're ready for him. And how do you do that? You have something bind to him, either a cofactor or a coenzyme. Coenzymes usually are organic molecules, and sometimes they are vitamins. Cofactors, usually metal ions like iron. Okay? And you see with this guy, this cofactor or coenzyme, doesn't matter, when he binds in here, he actually gets this thing and makes it a perfect fit. If he wasn't there, this molecule wouldn't fit there. So the holoenzyme is the apoenzyme plus a cofactor or coenzyme. Not all enzymes need a cofactor or coenzyme, but some do. So the ones that do are apoenzymes until they get their additional molecule, and then they're holoenzymes afterwards. We already said that. And some have to have these non-protein helpers. We talked about that. Some of these are RNA molecules called ribozymes. That's kind of interesting. Okay, enzymes more. So enzymes, we see there's a lot of things that come into play. One thing about these enzymes that will change them is temperature. If the temperature is too cold or too high, these enzymes will actually change shape, and they will be absolutely no good. Because as you can see, the substrate has to fit into that active site, which is where it has that conformational fit. This is why people die when they have too high of a fever. It actually causes our enzymes, since these guys are proteins, they denature. You've seen denatured proteins. You call them fried eggs, right? Those are proteins that are totally denatured, and they are not reversible. So that's why temperature is critical. Every animal has its own range. Those archaea that live in those 212-degree boiling springs. My enzymes and yours and most of the bacterias would totally get denatured. But these guys, the archaea that live there, have enzymes that are stable at that temperature. So that's just the thing, too high, too low, we can't do it, the bacteria can't do it, but we all don't have the exact same range. Some of them different. Here's another something that will cause the same type of denaturing as the pH. Too acidic or too alkaline is a quick kill for people or for bacteria. Again, there are some bacteria that can live in very acidic conditions, and especially some of the archaea, salt concentration, all that stuff. But there is a range, and for each specific bacteria, its enzymes have a range at which it will work. Out of that range, and it's no good. One of the main things, though, is going to depend on how fast things go, is how many enzymes do you have and how much of the substrate that you're working on. If you have a lot of enzymes and very few substrates, then things are going to go pretty fast. If you have very few enzymes and you have lots of substrate, it's going to go slower. Think about checking out at Wally World and there's only like 2 people checking out and you're in a big long line. It's going to go slow. But if they opened up all the registers, then we would all zip through there. And that's what we're saying about the concentration of enzyme and substrate. Another thing is the presence of inhibitors. And again, this can be a control mechanism for these guys. You get a competitive inhibitor that sometimes will block the active site on an enzyme, and if it's blocked, this guy can't get in. That's called competitive inhibition. It outcompetes this guy for those binding sites. Now, having said that, you could also have a non-competitive inhibition. We talked about cofactors and coenzymes turning them on. Sometimes these guys are always on, and if a inhibitor molecule binds there, it will actually turn them off, which in this case, you see this allosteric, that means it's not in the active site. It changes this guy's binding site so that this guy cannot bind, and that's the point. So sometimes we don't want our enzymes running full blast 24 hours a day. Sometimes we want them to normally be running and we only turn them off on special occasions. Sometimes we want them to be off most of the time and we only turn them on when we need them. These guys are just control mechanisms. So these guys will block those sites, but they're not going to denature the enzymes, which, as I said, is permanent. Denatured enzymes, you can unscramble an egg, guys. Okay, so there's the allosteric inhibition, where the inhibitor binds to the site, and of course we already saw that up there. The allosteric activation, where the binding site is normally not suitable to bind this guy, but when he binds there, it changes the active site and makes it suitable. We also have something called feedback inhibition. Some of you guys may not have had AMP2, so I won't refer to that. What happens is, once this guy, here, let me draw my own little thing. Now this guy doesn't have an allosteric site. What happens is, say I've got these 2 pieces individual. They bind in there, and when they bind, they become one piece. Well, what happens is this guy has got another side over here that when the levels of this raise up, when it binds here, it's going to act as a non-competitive inhibitor. That's what we're seeing here. Substrate is binding. Let's see. Yeah, let's go this way. Substrate binds to this guy. The enzyme does its job. Uh, then you get an intermediate substrate. It goes to a different enzyme, and you get chains like that. And then you get yet another intermediate. He goes to a third. Eventually, you take this guy and turn him into this. This guy is going to go up here, and when he binds there, he's going to shut down the pathway. And that's just a way of keeping from having way too many of these end products, because once you have enough, you've got enough, right? Most people won't go and try to buy out the fresh vegetable section of a grocery store. Why? Because the food will go bad before you can possibly eat it, unless you're feeding the entire neighborhood, right? So that's kind of the point on this, guys, is to keep the levels of this product down to the level that we want, and not to have a lot of excess, because it's a waste. Again, there's a functional protein, normal folding, and this one is denatured. And again, it's like taking your car key. If you take your car key and take one of those blow torches, and you can melt that guy down until he's a puddle of liquid metal, and then you can let him cool. You know what? You've got everything there that was there in your car key, but guess what? It's no longer the right shape. It won't open your car or crank it up. So the shape and if you think of these guys as being like keys, it's a lot easier to see why that won't work. All right, carbohydrate catabolism. As far as the eukaryotes, they're primarily going to do the same thing, except when it comes to the fermentation. Now, we know we do lactic acid, but a lot of these guys will actually do all kinds of things, including ethyl alcohol, which is the alcohol that people drink. Oxidize the carbs just like we do. Take the glucose, and the NAD comes in, grabs these guys. We make 2 ATPs. Pyruvate or something else. Ours goes to lactic acid. This can be anything. And then you take the CO2 off, and the 2 carbons go into the Krebs cycle. A lot more NAD, FADH, more ATP, electron transport, and you've got lots and lots of ATP. And you take that final electron and give it to oxygen. So glucose is going to be one of the most common carbohydrates used. It's a great energy molecule. That's why we use it. A lot of animals use it. A lot of microbes use it. The glucose, and again, you can do aerobic respiration, which is this whole part right here, or you can do just anaerobic, which is just that part right there. That's if you don't have oxygen, and the best you can do is like 2 ATP molecules per molecule of glucose, which is extraordinarily wasteful, because down here you can get over 30, closer to 40 actually. Okay, glycolysis, cytoplasm in most cells. You take 6 carbons that are arranged in a chain, or excuse me, not a chain, but a ring. You take those and you split those, and you have 3 carbon chains, 2 of them. You do that substrate level phosphorylation. Now, that's going to yield 1 ATP, or excuse me, it's going to yield 2 ATP for this one and 2 for that one, for a total of 4. But you had to add 2 ATPs, 1 there and 1 here. So if I had to spend 2 and I got 4, then my net profit is 2. And we get some NADH, but again, if you don't have any oxygen, that's not going to do a lot of good. And then you got that pyruvate, from pyruvate, you can change it into different organisms, change it into different chemicals. Ours is lactic acid. Some guys turn it into alcohol and all kinds of things. So the energy investment, the lysis, energy conservative stage. So there's our 6-carbon molecule. Here we are breaking it into 2, 3 carbons. And in the process, I told you we use 2 ATPs. There's one of the phosphates and there's the other. Okay? That's our energy investment phase. Now, we're going to send NAD in, and he's going to grab some hydrogens and take off with them. And what happens next on those guys depends on whether or not I have oxygen. Then I'm going to send 2 ADPs here, and I'm going to come up with 2 ATPs. What they don't show you guys is they've got another phosphate that's not attached to this guy. Well, it is. But in any case, there are 2 phosphates. We actually bind another phosphate on here beforehand, so that's why we can take the ATP. And then we wind up with pyruvate, and that's the end of glycolysis, the energy payoff. I don't think there's anything on there that I haven't already explained, but you guys can read that. Okay. Aerobic. What we do in aerobic catabolism, cellular respiration, is we take that pyruvate, that 3-carbon, and we oxidize it. We take hydrogen atoms, which have electrons. That's when we call it oxidation. And we make those into ATP by doing some redox reactions. So the 3 stages of this is we make acetyl-CoA, then we go into the Krebs cycle, and then the final thing is the electron transport chain. Here's our thing, and there's glycolysis there and where it comes. Now that pyruvate could come over this way, and here's our pyruvate. So we're going to lose one of the carbons, and we're going to match it up with 2 oxygens, and it's carbon dioxide. That's what we breathe out. When that happens, we've only got 2 carbons left, and that is acetate. So we take pyruvate, there's oxygen, and there's the carbon, and the hydrogen goes someplace. Well, it goes right there. So there's our CO2. Now we need a coenzyme called coenzyme A. Coenzyme A is going to grab onto these guys, or is going to actually bond with them, and it's going to be called acetyl-CoA. And then we have NAD come in and grab a hydrogen and take it to the electron transport chain. And there's our acetyl-coenzyme A. Okay, Krebs cycle. When we get finished, we're going to make 2 more substrate level phosphorylation of ATP. We're going to wind up with 2 FADHs, FADH2, sorry, and 6 NADHs, and we're going to make 4 CO2s. How am I going to get that out of that 2 carbon? Well, remember, the 1 glucose molecule, we're going to have 1 acetyl-CoA come in, and we've got the other half coming in after it. So each acetyl-CoA is going to give you half of that, but since we're running it twice. So there's our acetyl-CoA. This is a 4-carbon compound called oxaloacetate. So we add water, which, right, hydrolysis. We actually, the coenzyme A goes away, leaves that. So then we've got a 6-carbon molecule called citrate. It's going to do a slight change, and it's going to be isocitrate. Still going to be the same thing. It's just going to rearrange the furniture, so to speak. NAD comes in and grabs electrons, and then we take one of our CO2s and breathe it out. Okay, at this point we've got a 5-carbon molecule. alpha ketoglutarate. We're going to send in NAD. He's going to grab some more hydrogens and take it to the electron transport chain. Now we're also going to send coenzyme A in, and now that we're down, we've lost another CO2, so we're going to a 4-carbon. So this guy is going to be succinyl-CoA because we're putting coenzyme A again. Okay, at this point we can use either ADP or a GDP, and we're going to make a molecule by substrate level phosphorylation. When we do that, coenzyme A breaks off again. Now we're back to the 4 carbons. Aren't we back at the end? No, because we still have hydrogens attached here that we can harvest. So after this is succinate. FAD comes in and grabs 2 hydrogens. This guy becomes fumarate. We add hydrolysis, water in, becomes malate, still 4 carbons. NAD comes in one last time, grabs hydrogens, and when he finishes, we're back to oxaloacetate. Whew, that's a lot, isn't it? It's really not. You just have to spend time with this. Electron transport chain. Okay. So this is what's going to happen. NADH is going to come and drop this guy off. Let's see. Let me look at my thing. Okay. NADH is going to be oxidized back to NAD, where he can go and grab another electron. FMN is going to be reduced by grabbing the electron, and then he's going to pass that electron on to the next, and to the next, and to the next, and to the next, and to the next. So we're taking that now. We get rid of the hydrogen ions off of these. We've got something for them to do here in a minute. But what we're doing is we're stair-stepping the energy of that electron down. We're using that energy to pump out these hydrogen ions outside the membrane so that we can form a gradient. And eventually we're going to let these guys come back in, go down their concentration gradient, and when they come in, they're going to cause that enzyme to put that 3rd phosphate onto ADP. So all of these guys, they're series of redox. They get reduced and they get oxidized. And as they get oxidized, they reduce the one next to them. These enzymes just pass this guy down one at a time. Well, why can't we just do it all at one time? Well, when you release all of that energy at one time, we call that an explosion. So that wouldn't be too good for a living. So I think this is a better way to do it. That's just my opinion, but I think it works. So this is where most of the ATP is made, guys, both in humans and in the bacteria that are aerobic. Cytoplasmic membrane, the plasma membrane of the prokaryotes. Okay, here's another view of it. NADH dropping off, pumping this guy out, transferring the electron to the next enzyme in line, all these cytochromes, and eventually we take that electron after we've sucked all the energy we can, we add an oxygen atom, 2 hydrogens, and 2 electrons, which these guys are going to make just plain 2 hydrogens. 2 hydrogens plus 1 oxygen is water. And here we go. We're letting these guys come back in. The energy of their concentration gradient as they come in is used to take that phosphate and weld it onto ATP to be used for energy. 32, I've seen numbers from 32 to 38, so I won't ask you the numbers because I don't really think. It depends on who you ask, and it depends on the terms that you're using, so a lot, more than 30. That's our way of looking at it. Okay, if there is no oxygen, if you're in a low oxygen situation and you cannot do the Krebs cycle and the electron transport chain, then the only thing you can do is glycolysis. You get those two. And then you've got pyruvate, pyruvic acid. This is what we do. We send NADH and give it its hydrogen back, and it becomes lactate. Now, some organisms are going to take carbon, CO2 out, turn it into sorry guys, acetaldehyde. I don't know why I couldn't say that. I guess it's because I'm tired. But anyway, acetaldehyde is going to give its hydrogen back, and it becomes drinking alcohol. That's how they brew beer. That's how they distill whiskey. Is they use these yeast usually. But in any case, that's how we do this. That's called a fermentation. Lots of things are fermented. Sauerkraut, kimchi, all soy sauce. A lot of food is fermented. I think we talked about that earlier. So we have we need a constant source of NAD. Um, fermentation pathways. I guess if you don't have enough NAD to go around, you can shunt some of this over into glycolysis. Now for us, when we do get oxygen, we reverse this. We turn that lactate back into pyruvate, and then we send it back through to get the rest of the energy. All right. Yeah, there's the pyruvate. Lactic acid, which is what we do. It's also the way they make cheeses or cheddar cheese, yogurt, soy sauce, alcohol, acetone, and isopropanol, which is a rubbing alcohol. And this guy uses CO2 and propionic acid, Propionibacterium, and I guess that's the way they make that specific type of cheese called Swiss. So yes, Saccharomyces is a fungi. Those other yeast. Clostridium or bacteria. These guys I think are all bacteria. Aspergillus is not. Aspergillus is a fungus. Propionibacterium is obviously a bacteria. Well, I don't know why we got that guy there. Anyway, catabolism of lipids and proteins. Lipids and proteins look a lot like sugars. They're a bunch of carbons chained together. They've got hydrogens and oxygens, and there's all kinds of things. So these guys are really easy to convert into. You can convert these guys into a two-chain carbon. Hey, that is the acetate that we have, and then once we got the acetate, we just add the coenzyme A. We've got our acetyl-CoA, and we're ready to feed it through the Krebs cycle. These guys can be cut into a lot of precursors and go in at different parts of the Krebs cycle. So that's how we can burn fat. And also, in starvation states, human beings will burn the proteins. But bacteria can also, some of them, depending on the enzymes. If you don't have the right enzymes, you can't do this, guys. You got to have the right enzymes to break down these guys. But for human beings, they're all there. So there's glycerol, the 3-carbon, and there's the fatty acid chains. That's triglyceride. So if we add water here, we can actually break these bonds, hydrolysis, right? So we wind up with glycerol and 3 fatty acid chains. Well, glycerol is like DHAP, and it's a 3-carbon chain, guys. We can feed it right into glycolysis. And looky there, there's a phosphate because we got lipase has put ATP or used the energy to phosphorylate this, so that we can actually send it right on in. These fatty acids are easy to break up into 2 carbon chunks, and you can feed them right into to that. So there's the hydrolysis. Beta oxidation, well, there it is. We're just cutting these guys into 2 carbon chains, and I think that's everything I really want to say about that. Photosynthesis is just the ability of organisms to take light and carbon dioxide and water and turn it into glucose. And eventually they store this as starch, and we eat it as pizza and potatoes and so forth. Rice, all those kind of things that have starch. And all starches is just a long chain of glucose molecules. As far as photosynthesis, these guys have got special structures called chlorophylls, and you've heard of chlorophyll, the green pigment. These guys are sensitive to the light energy, and they can take that light energy and use it to bond carbons together. Remember we said that anabolism requires energy? This is what these guys do. The way the molecules are arranged in these guys, there's one right there. You can see it right there. Allow these guys to harvest that light energy and use that energy. These guys are in a cell membrane called thylakoids. In the prokaryotes, these guys are actually invaginations of the plasma membrane. So these guys sit here. And it's just the plasma membrane that just basically dives in and makes a little shallow or makes a pit for this to sit in. Eukaryotes, this guy is formed from the inner membrane of those chloroplasts, and then these guys are in stacks that we call grana. Stroma is the space between the outer membrane of the granum and the thylakoid membrane. So there's 2 types of photosystems, photosystem 1, PS1 and PS2. These guys absorb those energies, and then they use the redox to store energy as ATP and as NADPH. Remember, that's the one that we were talking about. These guys are light dependent, so they have to have light energy in order to have this. There are some that are light independent that allow these guys to make glucose from carbon dioxide and water without the benefit of sunlight. Biogeochemical cycles, carbon and nitrogen. So a lot of these chemicals move between living things and the non-living environment, and that's our biogeochemical cycle. Carbon between the heterotrophs and the autotrophs, these guys, the heterotrophs generally release CO2, especially the ones that do the aerobic cellular respiration through the atmosphere and then back to the autotrophs, who are going to use this along with water and sunlight to make food to go back to the heterotrophs. So the atmospheric CO2 is how this cycle works. So this is, if you've ever had a closed system, a terrarium, and that you can actually seal up, and it's self-sustaining. It has the animal and plant life that allow this to be self-contained, because this is the full cycle. Nitrogen cycle. there's a lot of nitrogen in the atmosphere, and plants definitely need this, but they can't use it directly from the atmosphere. So that has to go through the bacteria that transform it into different forms, and they're going to help the plants. Whew. Well, guys, we got it. It's a long slideshow, and it was a lot to it. Do look at the videos, do read the book, and do ask questions about this, okay? Because exam 2 is not too far away. We're in an 8-week semester. All the tests are close. All right, guys, I will see you in the next chapter.